So this part, anyways, as you see in terms of where the slides are, this is meant to be a recap on this 1D, 2D, 3D anyway. Let's keep it there. And again, we are going to discuss this issue of the quiz. 3D is very clear. You have, in three-dimensional space, a protein structure. That is the object that I show the pictures of. Here's a cartoon. That's the object that you can sort of look at. Well, you cannot look at it with a naked eye, but you can look at it by shooting X-ray beams on it. And that is defined by every single atom in this protein having a unique position in the three-dimensional space. So every atom has a coordinate it is assigned to. And that's what we refer to as the three-dimensional structure, because essentially from the information content, you need 3D coordinates to describe it. It's 3D. It's three-dimensional. What I show in this image here is on top of it you put the cartoon, on top of it you put some label. The arrows here, the beta strands, or these round things here, the alpha helices. But essentially this is 3D. 2D is essentially a distance map. So you have to put a protein here. This one has 99 residues. So from residue 1 to 99, 1 to 99. And every single position here is the distance of residue I to residue J in that protein. Black means they are close. Light means they are very far apart. The 2D information is one dimension less than 3D, but you can reconvert from 2D to 3D. So essentially it contains the same information. Then we go one reduction of a dimension further. We go, yes?
Does this distance take into consideration how the amino acid chain is folded? Or?
Yes, essentially that is, so the question is, does this distance take into account how the amino acid chain folds? Yes, so when the amino acid chain folds, it adopts this unique three-dimensional structure. So that you see here. And the distances that you show here are essentially for every single position here, you put yourself in one particular position, say 42 here, and you look at residue 42, and you look at all the other J's of the protein, you ask, how close are they? So yes, it takes into account how the protein folded. Essentially, that is exactly what you see in this distance map, and that is exactly why you can convert this from here to there. You can get the fold because it's actually written there, or it's put in there. Again, if you had made a mistake in this conversion, if you, for instance, had not put enough digits, a sufficient amount of digits, you'd just taken the integer, for instance, that may not be enough to recover the full 3D information. Okay? So you can go from here to there as long as you have the complete information without errors. And as long as it is complete. And yes, that takes completely into account how the protein looks. That is why you can go from this map, this distance map, into a 3D space. There was a question.
Yes, we can go from 2D to 3D and backwards without loss of information?
You can go from 2D to 3D and from 3D to 2D without losing information. Correct. Not completely correct. There is this only caveat. The mirror image. You cannot, when you go from here, from 2D to 3D, the image and its mirror, they look exactly the same in terms of the distance map. Because the mirror is just the rotation is the same thing. And that is a problem that you hope when you only have the two-dimensional information, that when you convert it to 3D, that you can actually figure out how the original, which one is the mirror and which one is right. So that you hope to get out, and that is a little piece of information you lose. But it's not really a dimensionality information. But there was a follow-up question from you.
When you have Q molecules?
When you have Q molecules? Yeah, chiral.
So you can superimpose, but if it's not chiral, you can't tell two molecules apart.
Well, again, so what you're looking at is you're bringing it to a single amino, to a single residue, right? But the point is that these are embedded in a chain. You cannot flip around in that chain. Okay? So that gives you that direction. That gives you that issue. There was another question. No? Yeah, two more here and there.
What's the deal with having a mirror image?
Why is it an issue with a mirror image? This is just for completeness.
Could that be a different protein? Or?
So does it actually happen in a protein that I have the protein in its mirror? No.
Where do you mean mirror?
So mirror is you completely turn it around. You look at the mirror, as simple as that. You look at the mirror, so you look at an image, or you look at a helix, and you look at its image in a mirror, right? And now you translate them to 2D. They give the same distance map. That's the issue. That means that when you come from the distance map, you cannot distinguish between the image and its mirror. Since proteins have sort of a directionality, most helices look in one direction, you are very certain that you can distinguish which one is the true protein. And in nature, exactly this does not happen. So proteins and their mirror images do not exist. Yes?
Yeah, so my question is, like, you told that we can convert it to 2D to 3D and 3D to 2D. So, and how can we make sure that when we are converting to 3D to 2D, there is no information now? Because generally, whenever we reduce a dimension, there is always some kind of...
So how can we make sure that there's no information lost? This is essentially has nothing to do with proteins. This is a feature of three-dimensional objects. You can completely transfer three-dimensional objects, any kind of three-dimensional object, as long as it is sort of a rigid three-dimensional object. It's not something that's totally dynamic. As long as it's a rigid three-dimensional object, you can always translate it into 2D and convert it back. And this issue with the mirror image is always the case. This is a general feature of three-dimensional objects. Okay, it has nothing to do with proteins. This reconversion is something that, again, it takes a lot of CPU. It's not that easy, but it's doable. Okay. The moment we go one further, we go from 2D to 1D. We also apparently reduce only one dimension, but we now lose very important information. We lose the information what is near one another. So one aspect of 1D is this sequence. The amino acid sequence, because it's a string. Think about it as a string. Any string is one-dimensional. Now, the secondary structure is also, although the word secondary suggests something else, and is often misunderstood to be 2D or something because of secondary, but ultimately you can write secondary structure as a string. For every single residue, you say it's either one of these strands here, these arrows, or a helix, or something else. So in that, you have a three-letter alphabet of secondary structure, and you can write that as a string, which you in this image here see where other is empty, is a blank space, and the E here is the arrow for that position. So the secondary structure is written as a string. In order to get the secondary structure from 3D structure, you need distance information. So you need more than 1D information. But essentially what you have as secondary structure, you can write it as a string, in that sense, is strictly 1D. There were two more questions, yeah?
That's like different from what is in Wikipedia, where it says like a three-dimensional form of local sequence.
Yes, that is different from a misunderstanding that many people have. That is correct. So he correctly observed that Wikipedia has a different way of looking at it. You are computer scientists. You are people who can think about it in terms of dimensionality. So the kinds of definition that on this issue that you may find in Wikipedia, and actually did not know that Wikipedia now took over these old books. This is sort of. This is an image that is 20 years old, and it was written in old books, and now Wikipedia sort of picks back images or ideas that were there 20 years ago and that are now, should have been dead already. Usually, I'm totally a fan of Wikipedia, but that is where Wikipedia goes wrong.
Secondary structure is one-dimensional.
Secondary structure is one-dimensional. And the primary sequence, as it is called, is also one-dimensional. It's from the information content the same thing. It's writable as a string.
And if we know enough about the domains that are in the secondary structure, we might also be able to rather losslessly convert to the other dimensions? Or?
So if we knew enough of a domain, so domain is a substructure, and that does not exist on this level of 1D, 2D, 3D. Domain is a subclassification. And in that sense, so subclassification could be, it's in this particular case it's not true, but it could be that here is a sort of local density, that would be a domain. And essentially you put a grouping to a bunch of 3D object features, right? And you could label this in 3D. You could simply say, well, these here You could point these here out on the distance map, and you could point these out here. That is a label, so it's not part of it. Now the question is, if we added that label, would that help us to go from 1D to 3D, or closer to 3D? And ultimately you're right onto something that I've been believing for a long time, and we still haven't put the proof out. But you could imagine that the more additional constraints you put, so you could have a bunch of 1D features that are such that if you combine them all, they allow you to recover some aspects of the three-dimensional structure. Maybe not the main, but maybe something, okay, I know this is a beta strand, and I know it is on the surface, and I know it's on the right side of the molecule or something like that. So you have a bunch of additional, all 1D features, but all taken together, you could actually say, okay, then it must look like that, the three-dimensional structure. And that is in fact what some people try. The proof is still out there. But in principle, yes, that is the direction you could sort of combine, hopefully combine. One way of doing that is deep learning combine. So put it into a machine learning device. And again, that is what people do, and that is partially behind some attempts from Google AlphaFold that work. Yes?
How does one read this 1D image?
I didn't hear what? How do you read this 1D image? I'm sorry. I should have clearly. So this is the sequence, and it runs from the first residue here, PQI. So those are the first 3 letters in the protein, okay? The protein runs in that direction. What I show here in the next columns are other proteins in other organisms, for instance. 1, 2, 3, 4, 5 other proteins. They are aligned. And this was supposed to be the topic of today, multiple sequence alignments. So essentially we see that there are these different organisms, proteins that look similar, and that for many places here the Q, the P have in fact the same amino acid, and for some they don't. Here you see there's a position, although overall they are very similar, there are places where not a single one of them has, for instance, oh, in this one there has the I, but not a single one has the T in this one here. Anyway, that is the way you read that. Then comes this column here of the secondary structure. And then comes something that is called solvent accessibility. Solvent accessibility essentially is with respect to the outside of the protein, how close are you to the core of the protein? Proteins, you can imagine, are spheres. But we will get back to that later. Did I answer your question? You started all of this. Do you feel? So tertiary structure is 3D.
And the 2D thing, is it also like the 2D model? What kind of structure is that?
So that would be called a distance map. Often people refer to it as a contact map. The difference between distance and contact essentially is if you put a threshold. You say that if a distance is below something, I call it a contact. So 2 residues are in contact if they're close enough. Okay?
Is it also like a tertiary structure, or is it like?
No, so that is 2D, and that's what I mean. So in terms of information content, you write it as a matrix. This one you cannot write as a matrix. Okay? This one you can write as a string, this you can write as a matrix, and this not. This you write as 3D coordinates.
3D is tertiary structure, secondary and primary structure is 1D, and what is 2D? Nothing.
It's the distance map. It's the contact.
But now
it does not really have another word. Distance map in itself is relevant. So there are 2 experimental methods by which you determine 3-dimensional structures. One is X-ray crystallography. You make a crystal of the protein, and then you shoot X-ray beams from synchrotrons. Not your normal variety of X-ray beams. You need a big machine for that. X-ray beams on it, and the scattering of these crystals allow you to sort of backtrace which residue is where. That is method number 1. And in fact, most structures today, 85%, are done by X-ray crystallography. The other method, other major method, is NMR spectroscopy, nuclear magnetic resonance. And there, essentially, you excite protons, and by the way they fall down, you put that in a magnet, and then you see where in space they are. And actually, what you get is distances. For every single residue, or for every single amino acid, you get constraints, how close in 3-dimensional space they are. So by this experimental method, this is essential to, these are essentially the experimental data that are used for these 15% of the proteins from an NMR, for which we know the 3-dimensional structure, to produce the 3D structure from experimental data. But there's no other word for that. Okay? Yes?
This 1D picture, we say it's 1D, but it actually has rows and columns. And we know that
No. But each object is only, so each object is only one string. I just, in this image that I show you several objects. And this way it becomes a matrix. But that's a different story. So this is one protein here. The next one is another protein in another species. I bring them close to each other because one of the essential parts of this lecture will be that this evolutionary information is relevant, that this multiple alignment information is relevant. So I will use this slide again and again. But each one of these strings is independent, or whatever I mean by the word independent. I'm not thinking about the matrix. Here, 2D, it must be the matrix. Without a matrix, you don't have a distance map. Distance map is defined as I to at least some subset of J. Yes?
So I think you said the experimental ways yield distance maps.
The NMR is one of the two methods that yields a distance map, yes?
Okay, so then, is that information complete, or do we have to guess at times?
So the experimental information from NMR is a little bit tricky. Is it complete, or do we have to guess at times? So the problem is that NMR gives you a constraint between residue, an arginine and an arginine. Now in a typical protein you have a lot of arginines, and the NMR method does not give you exactly which of these now do we have the constraints of. And that leads to a situation where you need absolutely a computer to find different solutions to the conversion from here to there. And then you need to make additional experiments, and then the experimentalist would not say that they need guessing, but it is not a one-stop go. It is not that you get this distance map from NMR, from an NMR spectroscope, and then tomorrow you have the 3-dimensional structure after some CPU time. But the typical process is that you get some preliminary NMR data, you throw a computer at it, after a few days you get some answer and you measure again. The computer tells you where is the highest uncertainty. So where should you get additional constraints, additional measurements, additional labels. That means you try to put a particular atom, like an iron or something that sort of gives you a label that allows you to see this one or that one. So you actually tamper with the protein sequence. So there's a lot of additional work. NMR is time consuming.
First experiment, afterwards we calculate, and then we need to experiment more to find out which candidate will be the expert.
Yes. So NMR is a little bit, so the problem of X-ray crystallography is really to get the crystal. NMR you can do essentially on a solvent of protein. And NMR, the problem really is to repeat the experiment often enough to gather enough experimental data. That is why also ultimately most of the data that exists is X-ray crystallography, and there are additional constraints. But I'm pushing this issue much further than I wanted. Yes?
So why is the distance given in kilocalories per mole?
Why is, sorry?
The unit is kilocalories by mole.
Oh God. Yes. Now, if you have an underlying energy model in the way you describe the proximity of residues and to translate that to an energy, that is the unit kilocal per mole here. Yeah. So the closer they are, the stronger they bind. So that's another thing that I actually, after 5 times giving that lecture, nobody ever asked this question. So another thing I have to wipe out. It's totally true. Thank you for asking it. You're totally right. I should just fake this and put distance there. Underlying is a program that actually computed the energy. That was the way to history. Let's not go into history. Okay, we close it on that one. We talked about dynamic programming, about the optimal solution. We talked about substitution matrix. This is very important. That's sort of the biophysical similarities between amino acids. Then we talked about a fast hashing method. And the major problem with that is you have to get the statistics right that allow you to sort of compute what's the probability of matching random, small, short random strings or words, and how far is your actual match from that away. I said that sequence alignment is something that is trivial as long as you're sort of what I dubbed to be the daylight zone, where everything is sort of simple, where you essentially any pair of sequences that you align, you know that they are similar, you know that they have similar three-dimensional structure. Then there's the twilight zone where the signal is beginning to fade. So some of those you have a similar structure, some of those do not have a similar structure. And then you get into midnight zone, where you can no longer tell whether two proteins have a similar 3D structure, because, for instance, they have 5% sequence identity, and 5% could be completely random. Yes?
What's on the x-axis here? Is that
the percentage sequence identity. So this one here is supposed to be percentage sequence identity. So the percentage of amino acids that are identical between two proteins. It's sort of a coarse-grained idea, and they are not clear lines here, and the numbers are not quite like that, but it gives you a rough idea. And what is important in this rough idea is the daylight zone is really the largest. The daylight zone is not such that there's all is trivial here, but there is a problem that is solvable. And the problem gets really complicated here because you fight against the growth of number. You try to find the one hit in 10 that is right, and that's really, really tricky. And you get into a place where we can absolutely not do it, full stop. So people try to intrude into this, but this is where we sort of can, with our best efforts, get in. And the relative proportion of these zones, that is correct. So while the precise number, is it now 40, is it 42, 40, 35 or whatever, that's not relevant. But what is relevant, most of it really in terms of percentage sequence identity, is daylight. And I showed you briefly that there's a phase transition here in the twilight zone. So in the twilight zone, things rapidly change. Yes?
In the midnight zone, there might still be similar structures, right?
Oh yes, and that is something that I do not talk about in this lecture. In fact, it's not only that there are many similar structures in the midnight zone. The reality is that if you look at all the possible pairs of similar structures, they all actually sit here. Or not all. The vast majority of similar structures actually have, sorry, this complicates it, and I didn't want to say that. Yes, there may be similar 3D structures in there.
Is it possible that because of like the evolutionary past that in the midnight zone, if you have like a similar structure, that it might it's more likely to be encoded differently?
Yes, so ultimately what we are talking about here, if we saw a pair of proteins that have, say, 2% sequence identity, and yet they have similar structures, how did that happen? Did it happen by divergent evolution? So we had two proteins in ancestor organisms, and they changed and changed and changed and changed. 98% of their residues changed, and now they have 2% identical, and they still have the same structure. And it's the same structure because they come from the same ancestor? Or is it that they actually come from no similarity? And again, so we have 20 amino acids, 100 by 20, average is 5, right? So the random, if you just randomly take two strings and you superpose them, is 5%. So getting 2% by random is totally trivial. So we would then argue, if we saw many of these, that at least the odds are high that many of those are convergent evolution, where convergent here really is convergent to the same structure. So we start from two strings that have nothing in common, and they not suddenly, there's an evolutionary process, right? Because that structure is stable, that structure gives you a function, that structure works, and that's why two completely different organisms sort of invent the same protein. Yes?
Alex, it's a bit different question, but eukaryotic cells are like the basis of all multicellular life, right? So
the prokaryotic.
Prokaryotic, right? Okay.
The prokaryotic.
They are that phase, okay. Did they evolve only once, or did they?
That is a totally cool argument. So so the question here is: did life essentially evolve only once? So if we believe that prokaryotes are the first sign of life,
Multicellular life.
Of eukaryotes? You mean ours? So did eukaryotes? So is there only one path towards the eukaryotes?
Towards multicellular life.
And that is something that I cannot answer because I really am not enough of an expert. It is hopelessly complicated. So there are two different types of bacteria. There are what people call the archaea bacteria and prokaryotic, or the normal bacteria. And there was a long discussion whether archaea is an independent branch on the evolution, and whether possibly eukaryotes came from these two branches. And with the latest data from sequencing over the last 20 years, people believe that really these are three different branches. So the answer is not given to your question. Are there two ways to eukaryotes? Are there two groups of eukaryotes? The current trees assume that there's one group of eukaryotes. And typically we assume that such a one tree exists if there is only one path toward it. But we do not know enough of evolution for that.
So basically all the life you can see with your eyes is kind of from one original cell.
That is one. So again, what is the precursor to? But now we are really losing our multiple alignment story. So what's the precursor to life? Ultimately, is the question here. And most likely the answer: this is pre-cell. So before a cell, life existed in small molecules of RNA, most likely, or life-like processes. Whatever formed life today began most likely as RNA, and that is before a cell wall exists. So at some point the cell comes. Now, how many different cells are formed at that point? I'm the wrong person to ask that. Can I take that as an excuse and move on to multiple sequence alignment? And if your answer were yes, or no, by asking further questions, I accept that. Okay, so there's this issue of pairwise alignments versus multiple alignments. And pairwise alignments, essentially, you take a pair of sequences and you sort of align, and you see that there is in this particular case, you would see that the best way of aligning them may be by inserting a point here. Now, if you look at the larger family, you would possibly see that actually the insertion should be before the two Gs. You would have a minor change because you see that there's a family. You see this pattern again and again. And maybe in the family, you see that for the family, the placement of where this insertion comes, whether it's the first G or the second G, makes a difference. I already showed you one example where this sort of thing is seen, where I said that when you observe something like that, again, this is a sequence of one organism running from the first residue to the last here. And the second, this is a sequence from a second protein, second species here. And wherever these lines are overlapping means they are similar. And then there are regions here where they are not similar. And then others here you saw are aligned only here, some are aligned here, and I said in the first lecture that ultimately shows you there's one and two domains, at least two domains in this particular case. But in this image you already see there's an advantage of not aligning only a pair of proteins, but many. Many give you ideas where it is important to conserve something. That is like motifs. Right? You get something where you see that, for instance, the simplest way of a motif would be you have a G, and in the next position you have an L or an I, and so forth. Right? So simple, regular expressions. That's the one way in which you think about motifs. And ultimately, the other way of thinking about it is just as a probability. So not a thing where you say L or I, but you give some probability for every single position. You have position -9, -8, blah, blah, blah. And then you have a certain probability, this is for nucleotide sequences here, you have a certain probability for having a T, a C, G, or A. And the height of the letter in this one means essentially that the probability of seeing a T here is highest, or the information content of the T is highest. That's why you can read only the T. In some cases you see positions here where you can read no letter, because essentially all four letters are equally likely. Okay? This is another way of writing a motif. It's the same thing like L or I in a probabilistic model, right? And Now, so in order to derive such a probabilistic model from bunch of sequences that you have aligned from a family of proteins, you want to build a probabilistic model, and the probabilistic model I will refer to, some people call it PSSM, position-specific scoring matrix. So for every single position, every single residue, you give a certain probability what amino acids you are likely to observe in that family at that position. And I refer to this also as a profile, and more often as a profile because it's just easier to say. It's all the same thing. It's essentially a bunch of probability numbers. So you replace the single 1D sequence by a matrix. A probability matrix that replaces, essentially instead of having a particular amino acid at one position, you now have a probability vector of 20, because you have 20 amino acids. Okay? If this is a binary vector, you're back to the simple string. And if you put values between 0 and 1 in their probability, then you have a profile. And the way you compute a profile, you start with a bunch of aligned sequences. You compute the log odds of this here. I mean, first you do the counts, then you do the log odds. So essentially you normalize by what's the background probability. How often are you going to see any of these amino acids by chance at that position? That gives you the log odds. And the log odds you then convert into a profile shown here. Again, what I showed you before here, the sequence is running in that direction from residue 1 to, in this particular case, 30 or 49 here. I show 4 sequences aligned in the family. This is sort of a consensus sequence. Let's not look at that one. But these are 4 different proteins in 4 different organisms, for instance. And then this alignment of these 4 shown here is explained by this log odd matrix, this profile, this PSSM. The idea is clear? Yeah?
Can you explain, please, because I didn't really understand.
So we begin with a sequence. We find all proteins that are similar to it. We build a family. So this family, in this particular case, has 4 members. And these 4 members are all linear sequences, all strings that we believe, by comparing the strings pairwise, they are similar, have all 4 of those have similar structures. Okay? So now we call it the family, because we have a bunch, 4, of things that have a similar structure. So they have something in common. That's why we call it a family. Okay? We could call it anything, except my name, family. And now I can simply go at every single point here, residue 1, 2, 3, say the residue 4, and I ask simply at position 4, at the 4th position of my string, what is it that I observe in that family? I observe 1 times k, 1 times e, 1 times a, and 1 times t, which is a high diversity, is as much as a diversity as with 4 sequences you can reach. As opposed, for instance, to position 3, where all 3 of them have a V. Okay? Now I simply look up this count, build a probability, convert the probability to log odds. At that point of the transition, from probability is just a count. So in the count here, I would say 100% is a V. In this point here, I would say 50% is L, 25% is S, and 25% is P. That's the simple count. And then I have to transfer this count to log odds. Log odds essentially means what's the probability of randomly putting an L there. And ultimately that has to do with how often do I see L in the database, or how often do I see L in that family. So there are different backgrounds models that you could construct. The simplest is really the entire database. Okay? So you essentially answer to the log odds answer to the question, how surprising is it that at that particular position I have 4 times V? How much of an outlier is that? And that is essentially captured in this matrix. That matrix is called the profile. The position-specific scoring matrix. Why is it position-specific? Because you remember there was this issue, we had the blossom matrix. the Blossom matrix changed from percentage identical amino acids to a situation where you assessed the biophysical similarity. Where we, for instance, said that matching an S on an S gives you a 2, while matching a W on a W gives you 17 in that score. So it's not only about matching identical residues, it's also about matching identical features. So some residues are charged, some are large, some are hydrophobic, some are hydrophilic, and you have to match these. And that's reflected in this matrix. But in this matrix, the matrix is always the same no matter which position of the protein you are. So W on W, whether W is at position 12 or at position 72, it will always, W on W will always be 17. This position specific matrix that I show here is changing that. Now I happen to not have a W here, but you could, for instance, look at the L at position 2 and the L here. And just the first number, the L on the A is 3 here, while the L on the A is 2. The L on the C here is -5, this is -2. So it depends whether you can change the L to a C or A, depends on which L we are talking about, the first or the last. Right? And that's why it's position specific. That's, yes?
So the profile lets you calculate the similarity from within your family, right?
Yes.
How do you get the profile in the first place?
So no, the profile doesn't let you compute the similarity of the family. The profile measures the similarity. So essentially, how do you get the family is the question. And that is, again, one way in which you could do that. I explained already to you pairwise alignments. And then I explained the hack to multiple alignments. So I do many pairwise alignments and I sort of merge them in a family. And that's the simplest way, and I'm going to show you a few. I wanted to show you a few methods that do it. I may never show you a few methods that do it. But there is a bunch of methods that usually in this lecture are presented. One is, for instance, called hidden Markov model. And that ultimately gives you these 4. Okay? Once you have these 4, you simply count at every single position. So the profile is a simple count out. Once you have them aligned, everything else is trivial. The problem is how to get it. And that ultimately would have been, in a different lecture of today, would have been the content of the lecture today. Let me repeat, since I will not get to methods that do multiple sequence alignments, that will not be part of the exam. Because the way this semester goes, if your questions hold the lecture, then I will just move on to the next topic, but you do not have to learn what we didn't cover, okay? Again, the videos are the crucial point of veracity, of what the truth is. What I cover in the lecture is what is going to be sampled in the exam, not what is on the slides. Okay, sorry, there are 2 questions. One was here somewhere. Yeah?
I think earlier.
Sorry.
In here we have consensus string from the family, I guess.
Yeah.
The upper part is aligned as an alphabetic.
This one here.
And I think in the consensus.
That is alphabetic. That should be alphabetic, yes, but it's alphabetic, yeah.
I think we are only looking third row for like consensus tree. That's why it doesn't matter at all which is like it's not matter. But how do you get the consensus?
How do we get the consensus? Again, I'd like to ignore the consensus here because the consensus. Just forget that there is a consensus. It's not relevant. We are no longer really using consensus. To think about it, the simplest way of thinking about it is you essentially pick the amino acid that you find in this composition that has the highest information content. So you pick the V because the V should be the most relevant. So E on E is 4, G on G is 4, V on V should give a higher number, 6. L on L gives 4, and that's why you pick the V. But again, forget about it. It's not really relevant. Yes?
I think W is only...
Oh, I'm losing questions. I remember now I forgot one here. There's another one there. Yes?
Like W is only in row 30, right? Like let's assume it would be only in row 30.
Yeah.
how would you know, like, for example, the score between L and W?
Where's the L come from? So again, you will get, even if in this count here, you have only four position, you have a probability of all the others, simply from randomly having a W. If you observe those four, but you could have had, right? So there's a random probability that essentially comes from the background model. You always have the background model on top. So what you see in the profile is essentially a mixture model, if you want, between the blossom and the actual observation in that family.
So you can use the blossom as a prior, basically, and then kind of
exactly
probabilities from the family you have.
So what you would use as a prior, as a background probability, well, one way is you could look at the database frequency. A better one would be you start with the blossom matrix, and then you see every profile is sort of a mix between these prior, the background model, and the actual observation. That's why, although you have, in this particular case, only V, you have a probability for all the other 19 matches. So let me go back. There was a question here. Still relevant? No longer? There was a question over there. Is it still?
Yeah, I was just wondering, so we get to this matrix on the right from the information on the left, so from the families, right?
Yes.
Okay, how exactly do we...
So how do we get from here to there? Again, I was too fast on this. Step number one, so it's explained on this slide in more detail, where the whole story is thought through for a much simpler alphabet with 4. Step number one, you just compute the counts. Convert counts. How often do you see a certain thing into a proportion, as a percentage at that position, or as a ratio? These are numbers between 0 and 1 that essentially count at every single position of the alignment here. This one, how often do you see each one of these symbols? That is here. Then you have to do a log odds translation. In this log odds translation, you need a background model. The word prior was used here. So essentially this is, what do you believe is a random match between A and C? And that random match you can get in a variety of ways. The simplest way would be, what's the percentage of C's in the database? Say if 5% of all the letters would be a C, then the random is a 5%. And that ultimately is what you put in as a factor here, and you get log odds from it. Clear?
So the numbers, just the position on the X.
Yes. Good. Yeah? No? Okay, let's get into position specific iteration for BLAST. So I presented BLAST as a fast hashing method that ultimately starts with words, matches words, then extends these words into longer alignments by using dynamic programming. And then in the next step, that's the dynamic programming part, so making it longer, and then there will be parts that are not done. Now, I said dynamic programming is nice, finds the optimal solution, but it's slow because it's in the square of the length, or essentially the length of one protein times the other length is its CPU time. And although that is not a lot for comparing a pair, for comparing many proteins against 150 million, this is a considerable time. And now I said that this extension here is done by dynamic programming. How do I gain from fast hashing if I use dynamic programming anyway? Why is this fast if I use dynamic programming? So I find words, that's fast as hashing, we understand that. But then I extend by using the dynamic programming again. So I pay. Why is it still fast? Yeah? Yes. But you're explaining why hashing is faster than dynamic programming. True. But what I'm asking is, now once we have the word match, once we have the hashing, we extend from the hash by dynamic programming. Why is that so much, why is that still fast? Why is that not a problem? Yeah?
Because we are pre-indexing, finding words and extending, and it's like indexing it, and we just add the amount of small.
The crucial part is this word small. Say you find one word in the middle of the protein, then you already split it in two. So since it's a square in the length, you quarter the CPU time. In this particular case here, you have 1, 2, 3, 4, 5 matches. Say they would be sort of roughly equally distributed, then you would sort of divide by a factor of 25. Because again, 1/5 square is 1/25, so it's a factor of 25. It's simply because the more words you find, the smaller the parts that you have to compare, and the shorter they are, the less problem the square of the length is. Right? That's the trick here. You break down the problem, divide and conquer, or divide and win through shortness. So the square of length doesn't matter anymore if the bits that you align are relatively short. And any breakdown helps you to reduce it tremendously. Again, the problem really is to compile the right statistics. Now, once I have the statistics, I simply can do the same thing again and again. So I can run the one protein against 150 million, and then I find a set of proteins that are similar. They're all pairwise similar. None of them uses this idea of a profile. But now I say have found 50 proteins. And now having these 50, so for each of those are such that I compare the one against the second, one against the third, one against the fourth, and. Okay? So I have pairwise comparisons that build up a bunch of 50 related proteins. And I can use them to compile a profile. I simply take the pairwise alignment. Okay? Now, since I know that the profile will help me to align, what should I do as the next step? Now I have a profile. What does that profile help me?
Find new members.
Exactly. So find new members is the answer, meaning that now instead of using the sequence with a blossom to find a new protein that is similar, I use this position-specific profile. And I say, so what I see in this particular profile, or what I try to show in this particular profile, there are 4 columns here that are purple, and these 4 columns are very well conserved, meaning that whatever you put, you need to, in the next sequence, if you're really similar to that family, first of all, you need to match these 4 residues. Okay? So now you're not going to just do a sequence pairwise alignment, you're going to use the profile against every single of these 150 million. In terms of CPU, it's the same thing as you just change the matrix. Previously, for every single position, you had a blossom matrix that is specific, position in specific. So the L on any other letter is the same wherever the L is. Now you have a position specific profile. And everything else of this step is exactly the same as before. So you
Memory, but it doesn't make no difference.
say again?
It takes more memory, right?
It takes a little bit more memory. The memory in this particular issue, the major memory is the database. And you're essentially using one, so the major memory is 150 million in the database. And now whether you use a string or string times 20 against the 150 million makes absolutely no difference. Yeah?
Does position specific mean that it has to, the match thing has to be at like after 1/4 of the whole protein? I mean, do they have to have the same length overall in order to match there then? Or?
No, so the proteins that are part of the family do not have to have the same overall length. Remember I showed you this example for domains where we have one protein that is very long, there was another one aligned, and then there were a few proteins here. So first of all, none of these proteins would have the same length as the first one. Only one would be sort of aligned throughout most of it. So that means that the profile would have more information content at some points than at some other points. So here it would be less informative because you actually have only 2 proteins that make up the family. Well, here you have 1, 2, 3, 4, 5 that make up the family. So this is more informative, this part of the profile. But essentially you compile the profile for the entire thing. Here you're back to pairwise comparisons because you don't have anything that is similar. Okay, this is like where you started from. And at some points, but this again is meaningful. Where this particular sequence is unique is part of what comes out of that. Yes?
Like this dynamic programming, right, is kind of a greedy approach. You first have to find like a word that matches and then
yes, dynamic.
And know how much you kind of lose on like matching by using this greedy approach, or is it like maybe you don't lose really anything, but it's always like something?
I'm not entirely sure. Are you asking would genetic programming work for that? Is that what I take?
Dynamic programming is like, it speeds things up, right?
Yes.
But at the cost that you might lose some points where there's not these words. How much is the percentage of stuff that you lose? Is it like an idea?
So dynamic programming you actually don't lose.
No.
Essentially, dynamic programming finds the optimal superposition given an objective function. So the objective function could be you optimize the percentage sequence identity. Given that objection, this objective, you can clearly get the top solution. There's nothing you overlook. Again, well, here's a caveat. The caveat is that position I and I plus 1 are independent of each other. The caveat is you cannot find, if you had an inverted sequence put in there, you would not find that inversion signal. And there are all kinds of things. But you will find the optimal superposition of 2 strings. And you will not lose anything. I misheard what you said. I thought you said genetic algorithm.
Oh, yeah. But don't you first look for like similar sequences, right?
Yes. But if your objective function is that the similarity of 2 strings is the goal of the thing, then you optimize exactly that function. Okay, it's sort of, I'm not talking about protein structures, I'm talking about similarity of strings. And that, of course, if you now said that dynamic programming may miss things because in structure things move around, or in evolution things change differently, that is true. So there are lots of things that can happen in evolution that I will miss with string comparisons. If that would have been your point, right.
Having just the sequence and even using maybe the blossom matrix or something, you get the optimum alignment even with like this.
You will get the optimal superposition of 2 strings, of pairwise alignments. And then from hacking pairwise alignments, you hope that you get the best multiple alignment. And again, that is not that simple, but this is all assuming that all you can do is comparing strings. If you had structure, you could do better. Anyway, so you iterate over that, you repeat, and that ultimately is the reason for the word psi-blast, a position-specific iteration of blast. So every single time, so the second time you run with the first profile, and then you get more sequences in the second round, you refine the profile, you run again and again until conversion. That ultimately is you run a sequence against profile. So you begin with comparisons of sequence against sequence, I call them pairwise, results is a profile, you use that profile and you run it against the database of sequences, and the result of that is an updated profile. That ultimately is what psi-blast does, and I'm going to flash. I just don't have the time. Again, flashing means that it will not be queried in the exam, but I still want to sort of run by you a few words of methods that use it and a quick idea. So that is what psi-blast does. Clustal, at this point, Clustal Omega is the latest version. Here is still Clustal W, Clustal X. It's a dynamic programming that starts, essentially doesn't use the database search. It uses a blast to find all the related sequences, and its objective is, once you know is those 1,000 sequences that we have to align, it tries to optimize this alignment of these thousands by dynamic programming. The dynamic programming is sort of the same hack that I showed you before, but in a much more refined way. With a cluster Omega W, you can sort of intrude into this twilight zone by using a sequence profile search, okay? And here's another method that does it. I'm getting over that. Another way is hidden Markov models. So hidden Markov models essentially are probabilistic models that have transition probabilities between states. And these states, I'm going to go over that. Whoops. So for alignments, essentially, what you do, you align two sequences. You have a deletion insertion state and you have a match state. This way you can essentially align two sequences, and you do that in the same way essentially as we did it, as I described it with a profile, but you do it with a so-called hidden Markov model. This is very prominent because it works very well. It's sort of an intrinsic way to compute the probabilities and to find the optimal path to the alignment of entire families. There is another way of doing it, the so-called genetic algorithm, that allows you, and I just run over it because going through details at this point is no longer possible. The advantage of the genetic algorithm is you sort of can find more combinations, you can put more information in there. For instance, one thing that you can do with the genetic algorithm, another thing that I really like Wikipedia, you can design this antenna here, and what may strike you as sort of a random fold is an optimal antenna designed by genetic algorithm, and you can also use that to align proteins. The beauty of that is you can have all kinds of additional constraints. So in all of these other models that I discussed, and all of what I discussed longer about PsyBlast, dynamic programming, they always use the assumption that I and I plus 1 are independent of each other in terms of the alignment process. While the genetic algorithm, you can completely drop this assumption, and you can put any additional constraint. For instance, the constraint being that for position I and I plus 2, I have to have a particular type of pair aligned. They must be hydrogen bond formers, or whatever you want to put in. Anyway, so this is a bunch of methods that allow you to go into the twilight zone. And now if we want to get a step deeper here, we use methods that do profile against profile. It's essentially, intellectually, this simple extension, right? Rather than looking at a sequence against a profile, you look at two profiles against each other. The problem with two profiles against each other is that ultimately it makes the comparison a little bit more complicated, because you have too many options. Now you essentially have two vectors, and you have to look at the product of two vectors. As you all know from simple geometry, there are two ways to compute the product of two vectors. So there are different ways of doing that, and that simple reality makes it more complex. But ultimately, these profile methods are used to go sort of deep into the twilight zone, touching on the midnight zone. Now, the most recent version of that, in fact, is a method called HS Blitz that also uses an alternative alphabet. So essentially computes or maps amino acid similarity or biophysical features into a new alphabet, and it uses that to be faster. I'm sorry for the rush through it. Yes? Thank you. So the question is, I said the profile is position-specific matrix. So I have a probability for every single position. But he correctly observed on many of these slides there was a gap. How can I have a gap and still a position-specific matrix? Think about the gap as amino acid 21. So this amino acid 21 has another probability. So intellectually, in terms of so I'm not talking about the evolutionary implication of how, but in terms of the computer implementation of how is trivial. I just have another symbol and I have a probability for another symbol, right?
My question is, I can't just put it on the list and calculate the possibilities, because I still have a lot of ways to move the gaps. So that it's not one matching.
Yes. You do have it at a particular position. So the gap comes at a particular position. Let me just look at a particular alignment. The gap comes at that particular position and not at that particular position. And that gives you a probability. Yes? And there's an overall count of gaps in your set of alignments, so that gives you priors. It is a little bit more fishy than that, a little bit more complicated, because a gap does not have a reality. 20 amino acids do have a reality, but in terms of mathematical processes, it's straightforward.